what I would want you to properly understand is why those research practices that are listed here are questionable. So why isn't it ideal that that researchers are doing this, are engaging in these practices? Which is what the Simmons article about, and which is what the next part of the lecture will be about as well. But first, I want to ask you a question. Why do we use statistical tests at all? Why do we use them in the first place? Why not just write down the results and say, hey, here's what happened. Good luck with it. I hope you know, right? Because you're currently enrolled in a course which asks you to do all types of flavors of ANOVAs and t-tests and regression analysis and what have you. So I hope you know where you're doing it. Anyone? Why are you doing statistical analyses?
Maybe to have a quantitative measure for actually standardizing, research, and so you can tell that you have a certain amount of people and you research a certain construct, and then you have a standardized measure of how you're gonna interpret these results, and in the end you can look at them and say, okay, there's a difference, or there's a significant effect or not.
Right. So if I'm understanding you correctly, thanks for this input. You're saying, well, we can use statistics, specifically inferential statistics, because not all statistics are about p-values and alphas and making inferences about populations on the basis of samples. But you're saying is that we can standardize that step, right? So
Yes.
we have some data collected, and then we can standardize to some extent the steps we're doing on the basis of that. So we're standardizing the inferences we're able to draw about the population on the basis of that sample,
Yes,
because we're using
I guess.
differential statistics. All right. I think that's a very good answer. I think it's a very good answer because that is indeed why we're using statistical tests, because we want to make an inference from the sample to the population. We want to conclude something about the population on the basis of our sample. Whether we want to and whether we can actually standardize those steps is another question, which we're going to cover in the next week's lecture. But researchers certainly have the desire for a standardized set of steps that they can take for drawing conclusions about the population on the basis of their sample. All right. Yeah. So I'm going to try and give you a simpler answer to this question, why use statistical tests at all. It might be to standardize those steps. It is indeed, you should know that it's related to making an inference, saying something about a population, a general effect on the basis of a specific sample. But then still the question remains, why are we doing that with p-values? Or what are we able to show with p-values and comparing p-values to alpha? And what you should know is that that central step exists, right? So we have a population, the whole set of potential observations that we could make, and we take a sample out of that, or sample observations, or a sample and the means that we measure there. And you find a significant difference between two groups in your sample, or you find a correlation in that sample. And the question then is, so what, right? Basically, you should imagine you as a researcher talking to people and say, well, I took a sample and then I found this, right? There's a difference, there's a correlation. And somebody goes, so what, right? Could just be random stuff happening because you took a sample out of the population. And that's basically what we're trying to, figure out, basically. We're kind of playing the devil's advocate and saying, well, if there was just random noise, how likely would these results then be? And that's basically it, playing the devil's advocate. Imagining yourself having specific research results and somebody saying, well, so what? And you're able to say, well, if there was nothing going on but randomness, this would be highly unlikely. So we're basically differentiating between three different explanations. So we find something, a difference or a correlation in our sample that we drew from the population of potential observations. And basically there's three types of explanations. So you could say there's just pure randomness going on, right? Because of all the random factors that took place in taking the sample or measuring responses from people, or just systematic variation, or perhaps a bit of both. So there's systematic differences going on as well as a lot of noise. That second explanation, so we're asking ourselves what the data generating process is, right? So we're interested as scientists to explain the difference I found. What could have produced it? Is it just randomness? Is it something else? That second explanation, that it would only be something else and no randomness at all taking place, no noise, no measurement error, for example, is highly unlikely. You would never observe that as a researcher. If you would encounter that in your data set, everyone in the experimental condition would score the exact same score, and everyone in the control condition would score exactly the same way. And I've never encountered that, right? So that would be very weird to have two conditions with identical scores in all participants. That's what just systematic variation would look like. So we can exclude that as a potential data generating process. What we still need to check for is whether just randomness is taking place. Assuming that there's nothing going on and the only differences we're seeing or correlations we're observing are just produced by randomness, right? And then we're taking that step to be the devil's advocate. We're saying, well, if it were the case that the null is true and all the stuff we're seeing is just noise, basically, then how likely is this test statistic, or at least this test statistic, or more extreme, if there were no difference in the population? So we're assuming if the null is true, how likely would this then be? So you're basically a bit playing the devil's advocate. Yeah, you're a researcher and you're saying that this process is taking place, but I think it's all just random. Okay, and you're allowed to say in that debate, the hypothetical debate, okay, if it were basically just there's no actual differences and we're just seeing basically noise, how likely is it then to observe this? And then you're able to say, well, if it were only noise, then this would be highly unlikely, right? So that's basically why you're calculating p-values, to be able to say that, to make that argument, saying, well, if it were just randomness, how likely would this be? But that's not the only thing we're doing, right? We're not only just looking at p-values and saying, well, something, interesting has occurred, because if there's actually nothing happening but randomness, this would be highly unlikely. So perhaps something is going on. That's what you do with p-values. You just check, well, if there's nothing going on, how likely would this be on the basis of that assumption? And you're done. That's what the p-value tells you. But we're using that in a decision, right? Yeah, what you talked about with Sharon and probably with Johnny in the first year, is that you're using that p-value and you're plugging that in a decision or a decision tree, the different steps that you need to make for rejecting or not rejecting null hypotheses. So you're using that p-value and you're comparing it with an alpha level, that 0.05 alpha level, that's kind of the default everywhere. And if it's below that threshold, you're saying, well, the p-value is smaller than 0.05, so it's significant and I can reject the null hypothesis, which is actually a form of practical reasoning, right? So we talked about practical reasoning in the first block, and I pointed out that making these types of decisions about either rejecting or not rejecting the null is a form of practical reasoning, as it was described in the critical thinking book, right? So you can list your alternative courses of action, in this case, rejecting the null or not rejecting the null. And then you can take into account different considerations. So what is the probability that I'm making a type 1 error? What's the probability of making a type 2 error? And what value does that have? What utility does it have for me to make these type of errors? How bad would the results be for me or society if a type 2 error occurred? Or how bad would it be for science or society if a type 1 error occurs? On a basis of those considerations, you're able to set your alpha level and your beta level and use that p-value in making those decisions. So you're comparing that p-value with an alpha level, which controls the type 1 error rate on a level you're comfortable with because of different considerations that you have, right? And you're designing your study and using a sample size that is adequate to show a particular type of effect, to gain a certain amount of power in your studies, to control your type 2 error rate as well. And then you're basically just making decisions. What you're actually saying is, well, we're striving for an alpha of 0.05 and a beta of 0.2 or a power of 0.80. Then what we're actually saying, the argument we're making is not only that devil's advocate, right, saying what has occurred is highly unlikely if actually the null hypothesis is true. No, but we're using that p-value and comparing it with an alpha level of 0.05. And what we're basically saying is that, well, if the p-value is lower than 0.05 and the power is at least 80%, then I should reject the null hypothesis. I've seen that the p-value is such and such, and I've seen that power is such and such, so I should reject the null hypothesis. What we're doing here is arguing that we should perform a certain behavior. We're saying, right, I can reject a null, or I cannot reject a null. So what we're arguing for is that our behavior is actually called for on the basis of the specific p-value we found and our considerations shown in the alpha and beta level. And that's a choice that you need to make as a researcher. Actually, it's not basically called for just saying we're using an alpha of 0.05 and a power of 0.80 just because everyone is doing so, right? That would be a common practice fallacy. Basically, what you need to ask yourself is, what are the consequences of type 1 and type 2 errors? And to what extent do I need to control for that, given the type of research I'm doing? But usually people go for the default values. My question to you is, do you think, or what do these default values tell us, right? Do people usually think that type 1 errors are more grave or type 2 errors are more grave? So what do we need to look out for more in our research? If we just look at these default values, think about substantive reasons why we should be on the lookout for type 1 errors, and a couple of them are mentioned in the Simmons article. For example, a type 1 error, so you're doing research, you're publishing results saying this is significant, this is actually happening, there's no noise, perhaps something is here. And on the basis of that, other people are going to do research as well. So a lot of research, time and resources are spent on continuing that line of research. And if that turns out to be incorrect, a false positive, then that would be a costly type of error to make. And indeed, that's why that could be one good reason for controlling type 1 error rates. As pointed out in the comments by Bram, you can also derive that from people's behavior, right? So if most researchers set alpha on 0.05 and beta on 0.2, what it tells you is that they're more concerned about making type 1 errors than type 2 errors, because the percentage of type 1 errors they allow is lower than the type of percentage of type 2 errors they allow in their decision procedure, because 0.05 is lower than 0.2. So we can actually derive from comparing the usual values for alpha and beta how concerned people are with type 1 and type 2 errors. And that default is what you take on as well. If you're just saying I'm going to use 0.05 as an alpha and 0.2 as a beta, then that's something that you inherit, right, from the majority in this case. All right, so we've looked at why we use inferential statistics. And we needed to go back to those basics, thinking about why are we doing stuff with statistics, with p-values, with alpha and beta in the first place. And we've seen two answers, right? So first answer is playing the devil's advocate, using that p-value to be able to say, well, if nothing was going on, this would be highly unlikely, basically, right? And we're doing a second thing as well. We're using that p-value and we're plugging it in a decision scheme, right? We're saying if the p-value is lower than a certain threshold, we are allowed to do something, in this case rejecting the null hypothesis, given that we've set alpha on a level we're comfortable with, and given that we have sufficient power to show the results we're interested in. So given that we've controlled type 2 error rates on a appropriate level as well. That decision procedure only works if you take all the steps appropriately, and because the decision procedure and the usefulness of the decision procedure is so sensitive to the extent to which researchers actually take the appropriate steps in setting up and conducting their study, we need to look at how those researchers' degree of freedom, those different questionable research practices that result from that, impact the usefulness of that alpha and beta, that decision procedure we just talked about. Doing that decision procedure and justifying your behavior in this way, right? Because if you look carefully, we're not actually concluding that the null hypothesis is true or false. No, we're concluding that we're justified in rejecting it or not. that this type of decision procedure is highly sensitive to the way in which we conduct research. So whether the type 1 error is actually 0.05, or the probability of the type 1 error is actually 0.05, depends on how many comparisons you make, and whether you correct the alpha level based on the type of the amount of comparisons you make. As we're going to see, people usually don't do that, so you can question the value of this decision procedure given the way in which a lot of researchers, at least in the past, have done their research. One way to think about this is the following. So if I were to ask you what are the odds of the outcome 20 when once throwing a single 20-sided die, these things actually exist. I think they use them for, I don't know, Dungeons or Dragons or stuff like that. And I would ask you, how likely is it? Well, so it's 1 in 20, right? So there's that 5%. And if I would then ask you, how likely would it be if you throw 100 dice, right? Then hopefully you would agree that the odds of 1 out of 100 dice showing the number 20 is more likely than one specific die showing the number 20 when you roll it once, right? That's just rolling more dice, having more chance experiments makes a certain outcome more likely than just having the one. And in a similar way, making multiple comparisons or having multiple dependent variables or multiple independent variables, but leaving stuff out, adding participants one at a time and checking the results, that's similar to adding more dice to your dice throwing situation, right? So you're actually rolling the dice with more dice, and that makes the probability of a fluke occurrence for the group of dice that you're throwing higher. So basically, you should be able to see why questionable research practices are questionable, because this is what people are actually saying. If I throw with a 20-sided die, then the probability to throw 20 is 5%. I throw a 20-sided die, then the probability of me throwing 20 is 5%. That's sound reasoning, right? You can say, yeah, that makes sense. That checks out. It's in line with the probabilities and the stuff you did. But if somebody would be saying, yeah, actually I threw 100 of those things, then you would be saying, no, you can't conclude that the probability is 5% of getting the number 20. That probability is much, much higher because you threw 100 dice. That's basically the same as what's happening in questionable research practices. So we're again taking a sample from a population or making an observation, which includes some element of random noise, perhaps because of random assignment. And that's that dice rolling, right? That's rolling the dice and saying, here's the chance part of the experiment. We're taking a sample. That means that we're just looking at a subsection of the whole of population of all possible observations. So what we're observing might be due to chance, right? And if we're then to make one specific comparison, let's say we make a comparison between one group and another group on one variable, let's check whether group A is more extroverted than the other group. Then you could indeed say, if we keep alpha on 0.05 for that specific comparison, the probability of a type 1 error is indeed 0.05. But if you're then saying, well, I'm not just making a comparison on one variable between two groups, but no, there's actually a third group, which I'm comparing with A and B as well. And I'm not just comparing on extroversion, but I'm also comparing on income, and I'm comparing on IQ, and you get a lot of more comparisons, right? So if you have a group extra and you have a variable extra, the amount of comparisons you can make multiplies as well. What then happens is the same as the situation with rolling the dice. It becomes more probable of getting a significant result on one of those variables, right? Because you're rolling those dice on, well, multiple comparisons. So basically saying, well, the probability of a fluke occurrence here is lower than getting one fluke occurrence in this group of comparisons here. And that's something we need to correct for. And if we fail to correct for that, that's a questionable research practice. Basically, what you're then saying is, it's still similar to the first argument, right? So only I translated rolling the dice to setting the alpha level. But the situation is the same. If you're making multiple comparisons between multiple variables and multiple groups, then the probability of a fluke occurrence, something that just happens by random chance, increases, and you need to correct for that. And if you don't, that's a questionable research practice. If you combine that with the temptation that researchers might feel, or be subconsciously drawn to publish significant results, that becomes problematic, right? So what might occur is called p-hacking. So going through your data in such a way that you will find significant results. So the likelihood of finding a significant result, if you compare multiple dependent variables, if you compare multiple groups, or you test a difference including a covariate, and then you take out the covariate and make the same comparison, see which one is significant and publish that one and leave out the rest, that is questionable because that vastly increases the probability of a type 1 error. The same applies to controlling or having a clear sampling plan. So if you were to say, well, I'm just going to do my data analysis, I'm going to check after each and every new participant whether I have found a significant difference. So you have two conditions, for example, you're checking whatever, and you add one participant to each group, and then you check the results. Another participant to each group, and you check the results. And for sure, somewhere along the way, you will get significant results, even if there is no real difference in the population. So even if the null is true, you will get a significant result somewhere. If you decide to stop only once you get a significant result, it's guaranteed to be the case that you will get a false positive result if the null is true. So the way in which you conduct your research, the way in which you go about making these comparisons has an actual impact on how useful that comparison between alpha and the p-value actually is. And it's actually very difficult to keep track of this. So it's easy to say, yeah, of course, when you add a different variable, you need to check for that. And it's sneaky if you don't, or you leave it out. But if you zoom out a bit and consider all the decisions researchers need to make from the start of having an idea about the research to writing the final sentence in their report, that's a lot of decisions, and there's a lot of forking paths, if you will, right? So what I just shown here is just a couple of decisions. So what I'm going to use as stimulus material and what I'm going to use as dependent measure makes for a lot of different situations. So let's say I'm doing research on the effect of wearing a lab coat on how participants view me. So you chose a lab coat in comparison with other types of coats, and then you measure that with a specific type of measurement instead of something else. What's difficult to realize as a researcher is that you ended up doing this and finding a difference, but what could have happened was something else entirely, right? You could have decided to use a different measure, or you could have dependent measure, or you could have chosen a different independent variable as well. What I'm trying to say here is that it's difficult to keep track of all the stuff you decided on as a researcher, and to bear in mind all the stuff that could have happened but didn't because you made another decision, right? And if you want to keep full track of the probabilities of different outcomes, including the probability of making a type of error, then you need to be able to keep track of all that, which is what it makes it so very confusing. This has been called the garden of forking paths. And basically it says that, well, you're walking through a garden, let's say it's a maze, you're taking all those turns, you're going left at A and you're going left again at B. And it's difficult when you've ended up at the end of the path, what turns you took, and to keep track of where you are and where you could have ended up with, where you could have ended up being if you made different decisions. What I'm trying to say is that we're not necessarily talking about cheating here, right? These questionable research practices are decisions that you will naturally have to make as a researcher. And sometimes it's obvious that you need to correct for something, right? So if you're saying, I have no clue about what results might occur if my theory is true, I have no specific predictions to test, I'm just going to measure a bunch of stuff on these three variables, then you should know that you need to correct for making multiple comparisons, because otherwise your alpha level won't equal that type 1 error rate. But a lot of decisions aren't so clear and aren't that consciously made, right? Why are you focusing on that variable, not something else? Why are you formulating that hypothesis and not something else? Why are you using that stimulus material? And why are you putting people in that condition and on in another, right? So those are difficult choices to make, and it's difficult to keep track of all the stuff that could have happened if you were to make different decisions at each intersection. All right. Just focusing on the questionable research practices that are mentioned in the Simmons article, researchers' degrees of freedom mentioned in the Simmons article, there's just 4 which they put in a simulation, and they checked how different the type 1 error rate, noted with the percentages here, would be from the implied significance level, noted above here, the alpha level, if you were to conduct if you were to perform one of or more of those questionable research practices. So if you were to include 2 dependent variables and not just one, and make a comparison on 2 dependent variables instead of one, right? So what I used as an example, you're not just checking extraversion levels, but also IQ, for example. Then if you're testing at an alpha of 0.05, the type 1 error rate of that decision procedure is actually 9.5%, almost double the alpha level that you would nominally hold on to. And what I did in the research, in the article, is run a simulation and check out whether doing those different things, so addition of 10 more observations per cell before checking the results again, controlling for gender or interaction of gender with treatment, and dropping 1 of 3 conditions, what impact that had by itself on the type 1 error rate. And you can see that the type 1 error rate will actually be higher than the alpha level if you conduct those questionable research practices. If you make combinations of the 4 practices listed above, then you get very high type 1 error rates. So even though those researchers would be testing at an alpha level of 0.05 for every comparison they're making, the probability of making a type 1 error, if you're saying, well, if one of those comparisons yield a significant difference, then my theory is confirmed, then the type 1 error rate for that collection of comparisons becomes very high, 60%, 61% even. So here we have our idealized procedure again, right? We're comparing p-values with alpha to control type 1 error rate and control type 2 error rate, and what we're seeing is that we're not controlling the type 1 error rate as well as we might have thought, right? The probability of type 1 error might be much, much higher than we're willing to have it. That might even be upwards of 60%. So if you're reading the research literature and you're bearing in mind, well, the odds are that, well, it's even more likely that this is a false positive than it is to be true, that changes your perspective of what you're reading, right? And that is essential, because usually researchers weren't that interested in having their results replicated, but we should be, right? If the probability of having a false positive result is that high, a better solution would be not necessarily controlling for every comparison you're making with a Bonferroni correction, for example, but especially since it's so difficult to spot all the decisions you made and all the stuff that could have happened but didn't because you made different decisions, right? It's difficult to keep track of the garden of forking paths. So it might even be a better course of action just to make sure that you replicate all research a lot, have a lot of direct replications. All right. So the argument becomes less convincing, right? So if the p-value is lower than 0.61, yeah, right. Am I really justified in rejecting the null? That's questionable. But we haven't looked at power yet, and we can do so because that's relevant as well. Especially if you're indeed going about replicating stuff, you should make sure you have enough power in your replication to show the original results. As you know, when we're talking about comparing p-value to alpha, we're just looking at one side of the decision tree, right? So we're looking at, given that the null is actually true, what is then the probability that we will still reject it? And we want to keep that type of error to 5%. But there's another type of error that you could make. It could also be the case that null hypothesis is not true, that there is an actual effect. And then we can look at a different type of error, failing to reject the null hypothesis even though it's not true. And we need to control for that as well. This is the ideal situation. You would control alpha at 5%, you would control beta at 20%, or you have power at 80%. But even there, the situation is different from what we would have ideally have it to be. So estimates for actual power achieved in most psychological research ranges from less than 50% or 0.5 to 0.35 in a more recent estimate, or even 0.31 in specific domains. So that 31% power estimate was from an analysis of different meta-analyses in neuroscience. So that's weird, right? That's paradoxical even. So most studies show a positive results. 96% of those studies show positive result, even though the power that we have for showing those results, if they were to exist, is fairly low. That's weird. So the situation becomes dire, right? So here we have our decision procedure, and instead of having a type 1 error rate of 5% and a power of type 2 error rate of 20% and a power of 80%, what we have is a power of 35% using one of those estimates, and an alpha of 61%. And a type 1 error rate of 61%. So that means that the probability of making those two types of errors is actually fairly high, and higher than we would want them to be, right? So our argument becomes even weaker. So if the p-value is lower than 0.61 and if power is at least 0.35, then I should reject the null hypothesis, right? That's not a very convincing way of going about arguing that you're justified in rejecting the null. It is, however, how people were able to show that that you would get younger by listening to the Beatles. So what they show in the article, and you should check up on that, is they show that, well, what we did, we actually compared 3 conditions, but we mentioned just 2 in the publication. We left one out. And we conducted optional stopping. So we gathered more data, and then we checked the results. We gathered more data, then we checked the results. We gathered more data, checked the results until we had a significant result. We had multiple dependent variables. We didn't report all of them, and we added a covariate, and we saw that the results were depending on the level of that covariate. So if we concluded it, we would show the results. If we left it out, it wouldn't show the results, but we didn't report that. So just like the Obama video, if you leave enough out, you get an interesting story. You get younger by listening to the Beatles. That doesn't actually happen. That doesn't actually happen. It's a fluke. It's dependent on a lot of type 1 errors. And it was easier to show, to get those type 1 errors by doing those questionable research practices listed below. All right. That also explains why Ben was able to show that stimuli from the future could influence the past. And we don't know that because Ben didn't go out and say, I did those questionable research practices. But Ben is very open in his reporting, and he was very open in the actual research article in the steps he took in analyzing his data. For example, he says that the experiments reported here required extensive pilot testing. As all research psychologists know, many procedures are tried and discarded during this process. And now, after the stuff we talked about, some bells should go off in your head, right? Oh, you've actually tried a lot of variations on this, right? And you discarded them because they didn't work, which means they didn't yield significant results. So please tell me about them. What did you do and what didn't work? And did you try 90 different ways of doing the research and just one turned out to work? 1 in 100, 1 in 200, right? So that's useful to know. It's all related to that garden of forking paths. What all roads we could have taken but didn't, right? And Bem is being very explicit about it. He's actually saying, well, this is what I'm doing. This is what a lot of people are doing. And I've tried out different stuff. I called them pilot studies because they didn't work, and I just published the stuff that did turn out to work. What he actually says as well about one of the studies that he did publish in the article is that in this specific study, the results from the last 50 sessions did not differ significantly from those obtained in the first 100 sessions, so all 150 sessions were combined for analysis. That shows that those questionable research practices over the degrees of freedom in action, right? He's actually saying, well, I've 50 additional sessions and I'm adding them to the first 100, right? So that's that whole optional stopping we're talking about. And he's explicit about it. He's saying, yeah, well, I gathered some more data and I just threw it together and did the analysis on that. What could have happened was perhaps the 50 by themselves being significant. So he would have published that, but it wasn't. So he went on and did another 100 sessions and added those to the first 50, and then it was significant, so you could publish that, right? So those are that's the Gardner forking paths in action. All right. It also is also how we can explain the brain activity in the dead salmon, right? Of course, the brain of, I'm not willing to accept the conclusion that dead fish show brain activity. I'm assuming that they don't. But engaging in dubious research activities could show false positive results, especially in neuroscience, because you're making a lot of comparison. So a three-dimensional pixel is called a voxel, in which you can use in neuroimaging. And there's actually a lot of flexibility, a lot of researcher degrees of freedom in setting those comparison levels, right? So I'm taking this cluster of voxels and comparing them to that cluster of voxels, and I'm making the groups of voxels smaller or bigger or smaller, and so there's potentially a huge number of comparisons that you can make on the basis of the same data set. So the probability of one of those comparisons showing a significant difference is substantial.